Again, but you all know of Neil Shubin and his book, Your Inner Fish, his work on Tiktaalik, and some of his work will be described today in his seminar, Major Transitions in Evolution, Fossils, Genes, and Embryos. Neil.
Thank you. In reading The Origin of Species, on the Origin of Species, Darwin's book, it's hard not to be struck by how much diverse lines of evidence are pulled together to support the notions of common descent, descent with modification, and natural selection. Facts of natural history, plant and animal breeding, paleontology, embryology, comparative anatomy, and so on. All are unified in a wonderful framework. But what's important about that framework is these different lines of evidence reinforce each other. They make sense of each other in a very meaningful way. They were formerly independent. Now they are linked in an understanding by our understanding of common descent and natural selection. And that's really been a theme of our field for the last 150 years is a synthesis of one kind or another. Every celebration of Darwin has its own synthesis. And the syntheses that we're dealing with today are increasing at a rapid rate. We're including whole new kinds of data that we couldn't have dreamed about as much as 10 years ago. And so that's really the theme of my talk today. Really, how do we pull together different lines of evidence to understand major transitions in the history of life? And what I am focusing on really is starting with expeditionary paleontology. That is looking at the fossil record as a predictable discovery tool where we can begin to assess and understand key moments in the history of life on Earth. That we can make discoveries that help us think about major transitions. But importantly, not only do they answer old questions, but they also provide us the ability to ask new and more profound questions. And that's really what it's all about, isn't it? I mean, being able to ask better and better questions with each new solution. And really what I'm dealing with here is the interplay between developmental genetics and expeditionary paleontology, and that they reinforce and illuminate reciprocally one another. And they do so because of the power of the tree of life and descent with modification. That was a mouthful, sorry. Anyway, so we will begin with, this is really what we're going to be looking at today is the origin of limbs, the transition from fins to limbs, the evolution of appendages. That's really been a topic I've been working on for the last 20 years. And what's interesting here is if you look at extant appendages, take the forelimb of a human or a chicken, or the fin of a lungfish or a paddlefish, they look utterly different. It really is hard to make comparisons among them or between these two kinds of things. These have fin rays and many bones, and these have a characteristic pattern of bones, one bone, two bones, little bones, and then the digits. And this calls for really suggests the importance of fossils in all of this, because as we add extinct taxa, we begin to see, and this is just a fraction of them, we begin to see forms that have both fin rays and that aspects of that one bone, two bone, further other bones pattern. And it's really these extinct fossil taxa from about 360 to 390 million years ago that tell us quite a bit about the intermediate stages in the evolution of vertebrate limbs. And I can actually blow this up for any of these other transformations here, and you can see sort of intermediate stages among many of these different appendages. But this suggests a very important and powerful role for paleontology. That is, what we can do to begin to answer these questions is to have expeditions that are targeted to key parts of the tree of life. And it's not random. We can actually use the fossil record, an understanding of phylogeny, an understanding of stratigraphy to predict likely places to answer our question. I'll give you an example of that today. But finding those fossils isn't the end point. It's the beginning. And it's the beginning of a powerful new way of asking questions, because oftentimes seeing these intermediate morphologies as recovered from the fossil record give me a new way to think about the developmental biology of extant taxa at the key points of the cladogram. That is, my understanding of fossils gives me new questions and new experiments to think about on extant taxa, and I'll give you an example of that. And then it's reciprocal. That is, my understanding of development and genetics gives me a new way of thinking about morphologies that I might have thought I understood before, and oftentimes suggest new areas or new problems to look at in the fossil record using our targeted expeditionary paleontology approach. All right, let's just deal with some examples here. I'll just run right through it, that reciprocal illumination. For me, it all began in Pennsylvania. I was born in Pennsylvania, but also I began my hunt for development for Devonian fishes in Pennsylvania. My first academic job was in Philadelphia. And what you see here is Pennsylvania is sort of festooned with Devonian age sediments. And they're of the upper Devonian age. And if you want to think about Pennsylvania 365 million years ago, get Pittsburgh, Harrisburg, and Philadelphia out of your brain and think Amazon Delta. Because what you had was an highlands to the east, an inland sea to the west, and a series of rivers draining from east to west. Now, if you're a paleontologist interested in finding fossils telling you about the transition from life in water to life on land, this is perfect because you can sample the ancient oceans, estuaries, all the way upstream, if you will. Pennsylvania had a problem in that it has very poor exposures for us to work on. Pennsylvania is not a desert. So my research with Ted Deschler, who was a graduate student at the time and since a long-term colleague, has been to follow the Pennsylvania Department of Transportation around as they put in new roads. So as they widen roads and put in new roads, what they do is expose cliff faces. This is awfully dark here. But what you have is a series of strata here, ancient streams, oxbows, meandering streams, and so forth, which produce fossils. And starting in 1990, we began picking the fossils out of there. And it was a really rich place to look because we found some of the earliest forests, this tree, Archaeopteris, numerous different kinds of fish, including these long, 16-foot long fish, armored fish, and early tetrapods. But there was one fossil that really changed my thinking and got me to think about new expeditions and so forth, and that was this one, which we discovered about 1 hour north of State College, Pennsylvania. It's a fin, an isolated fin. It has a shoulder girdle. It's clearly a fin because it has these fin rays right here. But inside these fin rays are the endochondral bones. These are the bones that compare to our own appendages. And it has a one-bone, two-bone pattern. And in fact, if you compare it to what we knew about Ichthyostega at the time, and I'm just doing this to force a comparison. This is actually a hind limb of Ichthyostega. This is a forelimb of Sauropteris. But you get to see that the pattern is approaching that of a tetrapod limb in a key way. One bone, two bones, two bones. Here you have one bone, two bones, two bones. And then you have rods that face away. Here they are flat and splay out in this bifurcate pattern, and here they're digits. But regardless, something about the pattern's the same. And it was really seeing those stunning similarities that got me thinking, boy, we really don't know a whole lot. And it's really time for us to think about looking at the fossil record in a different way, actually going back in time. Because when you map this on a cladogram, this is what we kind of knew at the time. We had a creature known as Acanthostega with an early appendage, limb with digits. You have lobe-fin fish down here. This is the one I just showed you, Sauropteris. But we really didn't know much about the limbs or even whole bodies of many of the sister groups of tetrapods. So to find the sister groups of tetrapods, really what we did is narrow the search to a critical window, both in space and in time. And for a variety of reasons I'll go into later, we ended up working in the Canadian Arctic. This is where our playground. We're about 600 miles south of the North Pole. This is Ellesmere Island here. Zoom into Ellesmere Island. This gives you an extent of the sort of vast exposures of Devonian age rocks. They were the absolute right age for us to find a sister group for tetrapods right here in this zone, the Fromm Formation. We've also actually now taken it lower in time. It's very hard to work there, as you can imagine. It's very remote, so we spend a lot of our time actually trying to figure out how to work there and get home safely. We get around in these airplanes, bush planes, which actually land on the tundra, and helicopters. So the logistics are difficult, meaning it's taken us a number of years to be successful. And it's kind of like the kids with the cookie. It took me 6 years to be successful with my cookie. We started off in 1999 in the western part of the Arctic, and this is what camp looked like. And it's a little dark, but these are the rocks we would work on. They're very flat. 1999 was remarkable for one thing. We didn't find anything of importance that year. And the reason for that was we were in pretty much marine rocks. So back to the drawing board, we went to the geological reconstruction, which was very similar to Pennsylvania, where we had an inland sea to the west and a series of rivers draining from east to west. The idea was to go upstream in the ancient environment, which in this case meant going east. And that we did in 2000, going to this eastern part of and the southern part of southern Ellesmere Island. And as soon as we did that, we started to find lobe-fin fish. Not the kind of ones we were looking for, but lobe-fin fish of a variety of types. It took about another year for us to find this site. This is the site the day it was discovered, before it was discovered. And what you see here is, I don't know if you can see it in this darkness, but here is, you see sort of a green layer there. That's a green layer because it's a carpet of fragments of bone right here. I mean, literally thousands upon thousands and thousands of fragments of bone that were weathering out of the rock. And it was found by our youngest field member, a college undergraduate. Anyway, so what we did was, this is us at 3 in the morning working that site the day he